Hello, I'm Barbara Tharp.
And I'm Deanne Erdman from Baylor College of Medicine Center for Educational Outreach.
Today we're here to talk about the third activity in the series, The Science of Microbes. It's called Magnifying and Observing Cells.
And as with all of our activities, they are aligned with the National Science Education Standards that go along with inquiry and life science. In this activity, students are going to learn that all organisms are composed of cells, which are the building blocks of life, and that almost all cells are microscopic and must be magnified to be observed.
Okay, I think we're going to start by looking at what do we need to do this activity. We need materials. And looking at the materials, we set it up so that we have trays of materials, one set for each 4 children or 4 students. This way we have an easy way to distribute the materials. We have the materials manager, which is one of the 4 jobs, come and pick up the materials, and then they're responsible for getting them back later. When we have the groups of 4, we usually have a principal investigator that basically kind of oversees the operation, whatever it might be. The exploration, we have a recorder-reporter who's in charge of making sure that things are recorded that they do. We have the maintenance director who is basically in charge of directing the maintenance. They don't have to do it all themselves, and that's really important to tell the maintenance director. With that in mind, the materials that we're going to use today include on the tray a piece of onion, glass slide, cover slip, some iodine, which we'll talk about in a second, tweezers, razor blade, which we may or may not want to have the students have access to, pond water, dropper.
And there's a plant in here, that the Elodes, or I've heard it called Elodes, either way.
Which is a common plant, waterweed, I think it's also called, and it's you can usually get that in the aquarium store.
And if you can't get this particular water plant in the aquarium store for this activity, you can use the thin leaves on celery, which you can always get at the grocery store.
Okay,
so that'll work. One other thing that's important that I have beside the tray is the material safety data sheet. And the material safety data sheet goes along with the iodine, and what it does is it tells you everything that you need to know about this particular chemical. It tells you whether you can dispose of it down, just something as simple as down the sink. It tells you what happens if you get it on your hands, if it's ingested, all of the safety precautions that you might need anytime you're using chemical, and that is something you definitely want to have at hand.
Where'd you get it?
It comes. You can see that this particular container, when it was shipped, has a material safety data sheet here. But before we came in to film this activity, I just went on the web and put in the particular chemical I was interested in and pulled one up. So they're widely available, very easy. And if it's something you're using all the time, laminate it so that you just have it in your folder to use each time.
Definitely.
All right.
Okay, I think next we want to make sure that we have consider all the safety that's important for this kind of a exploration. That includes making sure that the students follow all directions. And I have to say that with safety considerations, you want to make sure that these are set up ahead of time and that they're always followed. And it's nice to have them posted in the classroom as well. You might think about, like I said to begin with, students must follow all instructions. They must only begin when you tell them it's time to begin. That they aren't to taste or smell anything unless they want to waft it if you tell them it's okay to do that. They need to report any accidents or spills, make sure they're cleaned up quickly. They want to wash hands after, but sometimes it's a good idea to wash their hands before the investigation and after the investigation. And also how important it is to handle the cover slips very carefully, because at this point in time we're using glass. They do have plastic ones as well, but we do make sure that if it's glass, that it's taken care of, that if it's broken, it's disposed of properly, which means in its own container, carefully contained.
Okay, and just as we were talking a minute ago with the material safety data sheet, with this particular activity, the iodine certainly is toxic if it was ingested. If it gets on your skin, it doesn't hurt. It doesn't hurt anything. It will just have to wear off. If it gets on your clothing, the only thing that's going to take it out is a pair of scissors. So the parents aren't going to be very happy. But I will warn you that some people are very allergic to iodine. You've heard of sometimes people who can't eat shellfish. And that's a part of that iodine sensitivity. So if you do have anybody that has that sort of sensitivity, they would not want to be in contact with the iodine. All right, so to start out, we always like to focus the students' attention on what we're doing. And so we've got 2 photographs, and in the days of the overhead projector, you might have made a transparency of these, but you could use this slide for a PowerPoint slide to project, or you might even just find some similar images from the web that you want to put up. You may even want to make laminated copies of the photographs to pass around to students. There's a lot of ways you can handle this.
And we want them to explore and investigate, but if they don't have an idea of what they're looking for, then possibly a lot of times they're not going to find it. So we want to make sure that they understand what the Elode will look like when they see it, what the cell wall will look like, what the nucleus will look like, what the cell membrane. They want to make sure that they understand what those parts are, at least to some degree, so they know what they're looking for with both the onion and with the Elode.
You know, Barbara, that just made me think. This is the unit on microbes. Why are we looking at plant cells?
That's a really good question, but I think the idea is it's really difficult to look at one-cell organisms, but we can look at what is a cell by looking at a plant. And also, plants don't move around, and that makes it just a little bit easier to look at.
So the idea that just as we had an activity that focused on learning about magnification, we also are adding on to, in some ways, our knowledge bank about what it is that a cell is. If we're going to be looking at microscopic cellular one-celled organism, we better get a little idea of what a cell is before we get going here. So one of the things, just as Barbara said, they need some background knowledge, and so as you take a look at this diagram, you're going to want to introduce them to some structures that they're going to be able to see. And that's one reason that we chose these 2 particular materials, is that it is easy to see the nucleus. You can see a nice cell wall. The nucleus, of course, has all the hereditary information. The cell wall is for support, and all plant cells have those. The cytoplasm is everything that's between the cell wall and the nucleus, so it's all of the contents of the cell. And then, of course, the chloroplasts, which are involved in photosynthesis.
And they're those little green dots.
They are.
Cool.
They are. All right. So one of the things that we have is we have some student preparation cards so that the students can follow instructions for making their own slides. So the first slide that we're going to do, we're going to make a wet mount. And we learned how to do that in the past activity, but just in case, we kind of reviewed the steps. And so maybe you want to talk me through the first one. So we're starting with the onion, is that right?
Right. It's, the first thing you want to do is using a pipette or dropper, place 1 drop of water and iodine in the center of the slide. Let's just go with it in sequence here.
Here's some a container of water.
1 drop of water.
Okay, 1 drop of water.
And then a drop of iodine.
And a drop of iodine. And this is what I want to be careful with, so we're going to caution the students. You don't really want them to have a very big container of the iodine. 1 drop of iodine carefully. You notice that I have the slide on a paper towel just in case that we do have an accident here.
Okay, now it looks like what we have to do is we're going to get the onion skin. So when you think about what is the skin exactly, Deanne, what are we looking for here?
Well, when we take the different layers of onion, and really we used the razor blade or a scalpel or a knife earlier to cut some wedges like this, but when we look at the onion, you can pull off whole little sections.
I see, really.
And there's a very, very thin layer.
Look right there, you can get that edge.
Yeah, oh, there we go. There's a very, very thin layer that I can pull off, and that's just one layer of cells. And this is quite a big one. You could actually have a much smaller piece. But
she's going to place that
on the slide.
On the slide, and then you put the cover slip.
And then I'm going to put my cover slip
over the top of that.
There we go.
And it says when you put the cover slip on, you try not to squeeze any of the liquid out.
Right. I'm going to put it on.
She kind of puts it at an angle and drops it real carefully.
At an angle.
All right.
And just drop it down. And I may have to just barely tap it since this is such a large specimen. Then the next thing that I'm going to do is to put that on my microscope. And so I'll bring my microscope into view.
Okay, now we're going to put the onion slide on the stage, is that right?
And before I do that, I put it on the lowest power, because just as we learned earlier, when we're using the microscope, we always want to start and look at the largest area we can to get an idea of where we are and what this looks like. So we'll put it on. We'll use our coarse adjustment to bring the slide all the way up, and then I'm going to look into the eyepiece, and I'm going to focus it until I can see something that looks similar to the diagram that I'm looking at. You know, over the years of teaching, and I taught biology for a long time, I've had more drawings of water bubbles, the edges of cover slips, of everything imaginable other than what they were supposed to be looking at. And for some reason, when students start looking under the microscope, the first thing they get in focus, they just assume that that's what it is. And what that really points to, it just shows you how little experience they have, and how conceptually, how little understanding they have of what the microscopic thing that they're looking at really is. If they don't even recognize that they're supposed to be looking for something that's sort of oblong and rectangular, and they found something that's totally irregular and crumpled up looking, that they're not in the right area.
And that's why we had the pictures to begin with, because they need to know what they're looking for to begin with, right?
And so then I'm going to move it to the middle magnification and continue on with the process of looking. And probably with the onion, going to the middle magnification, or using the objective that's marked 10, and with the lens up here 100 magnification, it's probably going to be about as high as I need to go with the onion skin. All right, so what do we do next?
Okay, well, once they have got it in focus, then we want them to draw and observe and label the parts that they recognize, and record them. Make sure they record the magnification at which they observed it. And once they've done that with the onion skin, they've had a little experience now with making a wet mound, and so now we're going to go on to the Elodia or Elodia, the plant, and we're going to be able to make a slide with the plant. So I'm going to go through the directions again. It says, using a pipette or dropper, place 1 drop of water. No iodine, we don't need to stain this one.
All right, 1 drop of water.
Okay. Now we're going to take 1 small thin leaf.
1 small thin leaf, all right. I'm going to pull one off.
And you can use the forceps if you want to, or whatever.
Okay.
You're just so good at it, you don't need to use the forceps. Okay.
All right.
Okay, she's got 1 thin leaf. Now we're going to do the cover slip just like we did before. We're going to take it, pick it up, and lay it over there very carefully.
All right. So I've got my cover slip at an angle.
Okay. Much smaller specimen this time. Ought to be a lot harder to find.
Right. and then I'm going to go through the same process on the microscope. I'm going to put it on, and I'm going to again look for something that looks similar to the cells that we saw before. And one thing about the Elodia or Elodia leaf is that there's 2 layers,
which is really cool.
It is very cool, because when you're looking and on your microscope, if you'll remember when we were looking at the microscope before, you have the coarse adjustment, and you have the fine adjustment. And once I have it in focus, if I just barely go up and down with my fine adjustment, I can see depth and focus on different layers in my specimen. So the cool thing is that 1 layer of the cells are much larger than the other layer. So on 1 layer, you can fit 4 cells into the same amount of space.
It kind of frames the other one.
It kind of frames the other one. So it's kind of nice for the students to be able to see. Another thing that can happen with the Elodia is that chloroplasts do something called chloroplast streaming. And when you look at this, when you're thinking about this cell, you have to think of it kind of like a box. It's got dimension. Just because it's tiny and small doesn't mean that that it doesn't have depth. And so if you imagine this box, and in the center there's a big vacuole, and it's pressing the fluid and the cytoplasm against this firm cell wall, and it's making the plant stand up nice and rigid. And the chloroplast want to get close to the light source. So if the light source is up here, then they're going to make an orderly little streaming pathway around, and they'll actually begin to move. It's not really predictable about how to get it to do this. I've had the best luck by keeping the leaves in the dark and then bringing them out right at the class time, and it's kind of like, I don't know if they're waking up and going, oh boy, it's light, but the chloroplast many times will start to stream, and if one group gets them to stream, then everybody can go and take a look at it. But it's pretty cool to see that happening. All right, so we're going to do that and make our drawings as well, correct?
Okay, yes.
Right, and then after we finish doing this, we want to do some comparison and think about what we've seen. One thing I noticed when I'm looking at my drawings and I start to think about the size. Can I just right off the bat compare the sizes of the two things that
No, that's one of the things you think about comparison. You need to make sure that if you're comparing them at the same magnification. Because if you're not, then you're not seeing. They're not comparable. It's like having a giant car and a toy car.
That's right. So if I had one drawing that I made it was 100 power, and the other one was made at 400. And I said, well, they look the same size. They're very definitely not the same size. So you want to make sure that you think about, what the magnification was when you made the drawings, so when you're making that comparison. What about the overall shape? Are there some differences that you see?
Definitely. I hope they will see that, one is more, I think, more rectangular or close to square looking with the Elodea, and the onion is a kind of a long, flat, a little bit like a parallelogram kind of a shape, a long, thinner shape. So I get, for one thing, it's a neat way to see that cells don't look the same. They're cells, but they don't all have the same. Just like when we look at microorganisms in a minute, there are many types of microorganisms, but they certainly, and many of them are one cell, but that doesn't mean that one cell looks anything alike from one to the other.
Oh, absolutely. Well, and I noticed the onion didn't have any chloroplast in it, but it's a plant. I thought all plants had chloroplast for photosynthesis.
Don't they have to have green to have chloroplast?
I think so. And so that onion bulb, it's down below the dirt, the ground when it's growing. But have you ever seen the top of an onion? It's green.
So I wonder if we ever looked at that, if we would see any chloroplast up there.
I think we would. So we also saw a very clear nucleus in the onion cell, but they're not very visible in the Elodea, and part of the reason is that we didn't stain them.
Okay.
One of the things that it's important to talk about is to get the kids to start thinking about what these structures are and what they do, because as we talk about microbes, and we're going to realize that microbes or microorganisms, single-cell organisms, are very complex. They have to do everything that a full organism has to do. They have to undergo respiration, they have to get nutrition, they have to reproduce, they have to get water, they have to get rid of waste. And so we need to know what some of the parts are.
So they're like little machines.
Yes, and so you've probably heard. What's the control center of the cell?
The nucleus.
The nucleus. That's where the hereditary information is contained, and we find those in plant and animal cells, fungus, and in protists, where we have a very defined nucleus with a membrane around it. Then the cytoplasm. The cytoplasm is just everything between the cell membrane and the nucleus. So it's all the contents inside the cell.
And you mentioned something about that earlier. When you said that when a plant is standing up straight and tall, that's the cytoplasm holding it out like that, a part of it anyway. And so when the plants wilt, what's happening there?
Well, what happens is it's not so much the cytoplasm as that big vacuole in the center. And if you want to think about in a plant cell, right in the middle is a big vacuole that contains water. And vacuoles can contain other kinds of things, but there's a big central vacuole in a plant cell, and it's full of water, and it's sort of like putting a full balloon in the middle of that cell. It pushes everything firm against the outside, the cytoplasm, just as you were talking about. Now, if that plant doesn't get enough water, then the water content of the vacuole leaves. It starts shrinking just like a shriveled balloon, and it lets the sides of the walls semi-collapse, and the plant looks wilted.
Okay.
So
seen that a few times?
Absolutely, yeah. And then the chloroplasts, of course, are the green structures that are where photosynthesis takes place. And we talked about in the fact that even in the onion, while we don't see the chloroplast down in those cells of the bulb, we would see them up in the green stems. So kind of to recap, now that we've kind of gotten an idea of what a cell is, I think we're ready to start looking at some single-celled organisms and see if we can find see if we can find some of these same structures that we've been able to see so easily in the plant cell. Single-celled organisms, remember, are just as complex as a whole organism. In fact, in some ways more so because they have to do everything for themselves. So we'll be looking at some examples of organisms that are single-cell from the bacteria group, from the fungus group, and from the protist groups.
And you want to think about why microbes, or why is that even important? And I think it's amazing when you think about that the total mass of all individuals combined is more than the total mass of everything else on Earth.
Oh,
right?
Yeah, that's amazing.
That's amazing. And the number of species and the number of individuals as well.
And so to finish up, let's get our concept maps back out and see what else that we can add on that we've now learned as we're building towards trying to understand exactly what microbes are.
Thank you.